Hello everyone, I am Kate. I am an FSHD patient in the north of England, and I am here today to interview Julie Dumonceaux, who is a researcher at UCL. Julie, I'll let you introduce yourself now.
Yeah, thank you, Kate. So I'm Julie Dumonceaux, and I'm a professor at the Institute of Child Health at UCL, and I lead a small research group, mainly focusing on therapeutic approaches for FSHD.
I also like your background, Julie. Is it UCL? Yeah.
It is actually.
I'm very familiar with that part of the building. So, Julie, how and why did you get into FSHD research?
So actually it was by chance. At this time, it was like 15 years ago, I was working at Geneton and in France. And I attended a seminar, and someone presented a new paper talking about FSHD, and at this time it was, about FRG1, that is finally not one of the gene involved in FSHD, but I was interested by the seminar, so I decided to do some bibliography on FSHD, and I was amazed by the disease and how fascinating it was, and in terms of the genetic, in terms of the onset, etc. So I decided to really look at the papers, and then I decided to move forward on FSHD, and now I have a group working on this disease. So that's how I decided to move on FSHD.
Yeah. And can you provide an overview of the main research focus of your lab?
Yes, sure. So basically in the lab, we aim at developing some therapeutic approaches for FSHD. So we have different strategies ongoing, and we target the DOX4 mainly, that is the master gene of FSHD that is involved in FSHD onset and progression. And so we target DOX4 at DNA level, at mRNA level, and at protein level. So for the DNA level, we already published a paper using the TALEN CRISPR technology, and we have shown that we are able to cut the region on D4, and when we cut this region, we inhibit the production of DOX4. So that's one possibility. And when we also target DOX4 at mRNA level using antisense oligonucleotides targeting the mRNA, and in this case, the antisense oligonucleotides binds the mRNA and induce its and destroy the mRNA. So if there is no mRNA, there is no protein. And we inject antisense oligonucleotides, or we vectorize this antisense oligonucleotide using an AAV vector. So in this case, the antisense oligonucleotide is produced in the cells after the injection of the AAV vector. And we also develop another strategy at protein level. That's the decoy strategy. And in this one, we produce a decoy that is mimic the DOX4 binding sites. So the decoy is able to bind the DOX4 protein. And when the decoy is bound to the protein, then DOX4 is trapped and is not able to bind the genomic DNA anymore. So DOX4 is still present, but cannot bind the DNA, so it cannot induce the misregulation of all the genes that are the DOX4 nectar genes. And finally, we develop also a biomarker of the efficacy of the therapy we develop, and that's a very, very important point, because so far we do not have reliable biomarker that can be used in clinical trials, for example. So this is very, very important. And one of the biomarkers we are looking at is the myostatin, because myostatin is produced by the muscle. And for us, it's an indicator of the muscle health. When we have a low level of myostatin, this means that the muscle is not really in good shape. And when the muscle feels better, the myostatin level increase. And so when we look at the myostatin level, we can say whether or not the muscle feels better after the administration of the therapy.
Right.
Basically, we do on FSHD.
Yeah. And I've also read that your lab's done a lot of work into the poly-A signal. Can you explain what that is and what the significance of the poly-A signal is in FSHD?
Yeah. So poly-A, that's the polyadenylation signal, which is a 6-nucleotide sequence that is present in all of the messenger RNA. And this is a particular sequence that is absolutely essential for the mRNA stabilization and the mRNA nuclear export. So when there is no polyadenylation signal, the mRNA is unstable and is degraded, and the mRNA will not produce a protein. So if we target the polyadenylation signal of DUX4, our expectation is that without the polyadenylation signal, the DUX4 mRNA is destabilized and there is no production of the DUX4 protein. So we decided to target the DUX4 polyadenylation signal because it was shown that individual carrying mutation, so the contraction of the D4Z4 repeats located on chromosome 10, because in the human genome, there is 100 of location of the D4Z4 repeats. And one of the most important is on the chromosome 4, and that's how FSHD1 patient is characterized, because they have this contraction of the D4Z4 array on chromosome 4. But very interestingly, when this contraction occurs on chromosome 10, this is not associated with FSHD. And actually, it was shown that it's because the polyadenylation signal of DUX4, which is outside of the contraction, which is on the subtelomeric part of the chromosome, is mutated on the chromosome 10 compared to the chromosome 4. So that was very important to show the crucial role of the polyadenylation signal in FSHD onset. So that's one of the reasons we decided to target the DUX4, the polyadenylation signal of DUX4, because it was shown that when the polyadenylation signal is mutated, this no longer leads to the production of the DUX4 protein. And second, the second reason is the fact that in the testis, it was shown that DUX4 was produced. But the exon 3, which carries the polyadenylation signal that is used in the muscle, is not used in the testis. And another polyadenylation signal is used in the testis that is located in exon 6, 7, or 8. So if we target the leucine and antisense oligonucleotides, the polyadenylation signal located on exon 3, we target only the mRNA that is produced from the muscles, and not the mRNA that is produced in the testes, and that which could play a natural role in human. So that's the two reasons why we decided to target the polyadenylation signal of DUX4.
Right. So is the poly-A signal then significant in both FSHD1 and FSHD2?
Absolutely. Right. Because the production of DUX4 is the same in FSHD1 and FSHD2 patients. Yeah, there is no difference.
Okay. And as we have started a new year, could you tell me what the most significant development in your lab last year was?
In my lab?
Yeah.
So we develop therapeutic approaches for FSHD, and we show that one pathway involved in cell death is misregulated in DUX4 expressing cells. That's the necroptosis. And so I think that's the most important result we had last year, and we published this few months ago. And we have shown that necroptosis is involved in FSHD and in DOX4 mediated cell death. So necroptosis is a new target, is a new therapeutic target.
Right, fantastic. And so in 2022, are you planning on continuing with that research?
Yes, of course, we're gonna continue that, and we have a funding from the FSHD Society to do so, and we are actually recruiting a postdoc to perform this experiment to go further on the role of necroptosis in FSHD and in DOX4 expressing cells. But we also continue our other research concerning the antisense oligonucleotide and the decoy mainly.
Fantastic. And as we sort of move towards getting clinical trials, hopefully in the near future, what's your feeling about the chances of treating or curing FSHD?
Yeah, so that's not an easy
the impossible question.
That's not an that's not an easy question. So curing mean that you totally you totally cure someone from the disease, but if you treat, so there is some improvement. So my feeling is that to date a cure is not possible, but treating patient might be possible. And I think that the most promising thing is to at least stop the disease progression. Because the point is that in FSHD, it's also the case in many muscle disease, you have an atrophy of the muscle fiber and the loss of the muscle fiber. So one important thing is to stop this loss of muscle fiber in order to maintain the force and to maintain, you know, the ability to walk, etc. But another thing is to rebuild the muscle. And so one possibility to do so is to play with the myostatin, but it's not an easy way to do it because so far the results have been, you know, disappointing in patients. But I think that first we should try to at least stop the disease progression, and then when the disease progression is stopped, we can think about rebuild some muscle fibers.
Yeah. And as you alluded to that, it's very difficult to deliver drugs into the muscle. Have there been any promising advancements that will make that easier in the future?
I think so. So there are many labs working on the delivery of different drugs and different conditions to improve this. So, for example, when we talk about antisense oligonucleotides, different conjugates have been published, different, you know, cell-penetrating peptide. Now people are moving on antibodies, so you can combine an antisense oligonucleotide with an antibody that will recognize the receptor on the surface of the muscle fiber, and this can improve the uptake of the antisense oligonucleotide by the muscle fiber. So different companies are working using this kind of antibody conjugated antisense oligonucleotide or siRNA or, you know, this kind of things. A new field is the nanoparticles, and so there are new lipid nanoparticles that are used, and they also may improve the uptake of the antisense oligonucleotide by the muscle fiber. And another possibility is to also modify for the people using AAV vector. There are several papers showing that you can, you know, engineer the AAV vector in order to improve their efficacy. So if you want to do so, you have several possibilities. You can play with the capsid, and there was a very nice paper published in Cell like few months ago, showing that by modifying few codons on the AAV capsid, you can massively increase the targeting of the muscle and decrease also the targeting of the liver, which is very important for AAV. So you can do that, or you can also increase the expression level of the genes you want to express. And if you have a low, well, not a low, but let's say a delivery, and you can't improve the delivery, what you can do is to improve the expression of the gene you delivered.
Right.
So that's also one possibility. So I think it's still an issue.
Yeah.
Because we see that for the other diseases such as MTM1 or DMD, pharmas are increasing the quantity of AAV vector they inject, and we see that there are some limitations to that also.
But they're essentially trying to flood the system. Yeah. Yeah.
Yeah.
Okay, which is not ideal, I'm guessing.
No, which is not ideal. So we know that we have to improve the uptake efficacy. Yeah.
Yeah. So it's still a major challenge to be overcome. Yeah.
It's still, but it's ongoing. I'm pretty confident about that.
Yeah, right. That's good.
Yeah.
Yeah. I guess it's also one of those things that affects a lot of different diseases. So there's not just the FSHD field working on it.
No, absolutely not. Yeah. AAV vectors are used in many muscle diseases. So and they're more advanced than us. For if you look at DMD or as I said, MTM1 or SMA. They've been using AAV vector for a very long time, and we can also look at the clinical trials that have been already made for these diseases and learn many things. So by looking at the others, we can improve the therapy for FSHD.
Yeah, and generally, are you hopeful that you will be able to get something to the clinic anytime in the next decade?
I hope so. In the next decade, I'm pretty confident. We have really nice collaboration with a researcher for example, the combination of the antisense oligonucleotide with CPP. We have other collaboration with people working in other, nanoparticle fields, this kind of thing. So yeah, I am pretty confident that we will be able to deliver our antisense very nicely in mice to begin, and in human.
Eventually.
Yeah.
If I put a mouse costume on, does that count?
What?
If I put a mouse costume on, does that count? Can I be a mouse?
Yeah.
So as an academic research lab, what are the biggest challenges that you face in trying to get into the clinic?
Well, that's always the same response. That's money, because everything is really expensive. We have no idea how expensive it is. When we start using animals, it's really a massive amount of money. For example, to maintain the colony we have, it's around 15,000 pounds per year. So it's quite expensive. And then you have the salary of people, which is also very important. But because I'm in the UK, I think that I'm facing something also different from the other countries. It's at the Brexit, which impacts our research in different ways. First, we have to pay fees when we order things. So it's so the cost increased, okay, for everything. That's the first point. And the second one is that, if you want to come to work in the UK, if you're a European, now you need a visa. So it's less easy to do, and we have position open, and we have difficulties to recruit people because I think of the Brexit and of course of the COVID. But I think that's the main reason is the Brexit, even if I can't prove it, of course.
And so as a former UCL student, on a more positive note, I'm interested to know what is the best thing about working at UCL, even if we're not in the EU anymore.
I think that's, well, two things. The first one is the international reputation. So when you say that you work at UCL, it's everybody, you know, say, wow, that's really good. And so I think it's a good thing, because when you ask for money, because you work at UCL, it's better. For sure. That's the first thing. And the second one is the excellence of research. People around me are really good. We have also a very good collaboration with some groups like Francesco Montoni, okay, which is a leader group in the muscle field. So they're top researcher. And it's very impressive and very, you know, interesting to talk with people around me, because they're really good. So yeah.
Yeah. And what do you think are the chances for FSHD clinical trials to come to the UK? What hurdles do you think we need to overcome as a country to actually get patients on trials?
Yeah. Maybe you are referring to the fact that the fulcrum trials were not launched in the UK during the first phase. So we are aware that Europe and UK are not as advanced and organized compared to the USA. So actually, there are two important initiatives that occurred last year. The first one is the FSHD European Trial Network. That's a working group working for trial readiness in Europe. Okay, and the idea with this group, with this consortium, is to prepare Europe for clinical trial for FSHD, by example, by attracting FSHD clinicians, because there is not enough clinicians for FSHD, and also to identify potential centers for clinical trials and to assess their readiness and willingness. That's very important. So that's the role of this FSHD clinical trial network. But there is another group that is exclusively, so this is an European group. The UK is involved, but also France, also Netherlands, Italy, Spain, etc. So it's a very important group. But there is also a UK specific initiative that was launched by Raj Bajani, that's the UK FSHD group. And the idea is also to get UK involvement in clinical trials. So there are different groups in this UK FSHD group that are, one group works on how to bring more physicians in the FSHD field. Another group is on research. How can we collaborate more efficiently, etc. So it's a very, very important. These two initiatives are very, very important, and I think that especially for the UK. Yeah,
and what do you think that patients and families in the UK can do to try and help with those initiatives?
Yeah, I think that it's important to have the voice of the patients. What are the patient's expectations? What do you expect from us, from the research? That's very important, and also that's very important to be involved in, for example, EMA, European Medical Agency work group, because there are meetings regularly, and patients are involved in this meeting because they can say what they expect, etc., and they can speed up the process to go to the clinical trials.
Yeah,
that's very important. If patients are involved, it's very, very important. It's absolutely crucial, actually, the involvement of patients in the research, in this kind of initiative, and EMEA. That's very, very important.
Yeah, and natural history studies as well.
Yeah.
Yeah. So
absolutely. Yeah, register. So it's very important that the patients also register themselves in the UK patient register, UK or other, but patient registry.
Yeah, absolutely. So we still obviously have a lot to learn about FSHD. In your opinion, what is the biggest gap in the knowledge at the moment that needs to be overcome?
Yeah, so I think that's not an easy question, but maybe to really understand the role of DUX4 in the disease onset of progression. So for the moment, we know that DUX4 is involved. Okay, there are many evidence showing that DUX4 is involved. DUX4 is expressed in FSHD1 and FSHD2 patients. The methylation drives DUX4 expression, and there is a loss of the repressive epigenetic marks in the FSHD patient, and this loss leads to DUX4 expression. The 4QA allele is required to declare FSHD, and the 4QA allele carries the polyadenylation site of DUX4. So we have many evidence showing that DUX4 is involved, but actually we know that DUX4 drives different, impair different function, impair the muscle function, induce the muscle fiber death, increase the sensitivity to oxidative stress, etc. But we don't really know what exactly is the role of DUX4, and what in all this potential role of DUX4, what is the one which is the most important to drive FSHD onset. Because if you look at DUX4 expression, it's quite surprising, because DUX4 is expressed at very, very low level in one nuclei, in 1,000 nuclei. So that's very, very low. There is an expression of DUX4 in the fetus, in FSHD fetus, but you'll see the effect of DUX4 expression 10 or 20 years later, or maybe, or sometimes 50 years later. So it's really surprising, and so it's hard to understand how such a rare protein is able to drive the disease, and how. So I think that's a key question. That's still a key question in FSHD. That's the exact role of DUX4 in the FSHD onset.
Yeah, one thing I've always wondered as well is why is it that DUX4 only affects the skeletal muscle system and not the other muscles of the body?
Because DUX4 is expressed, is expressed only in skeletal muscle. You can see a rare expression in some other organs, or a very, very low level, but actually it was published by the Peter group years ago now, that you have a myogenic specific enhancer, meaning that the expression is driven by a myogenic protein. So this protein are only expressed in muscle, and that's why the expression of DUX4 is restricted to the skeletal muscle.
Right, fantastic, thank you. And then some funner questions. Which scientist, living or dead, has been your biggest inspiration, and how have they influenced the work you're doing now and your approach towards treating FSHD?
Well, okay, that's for me. That's not an easy question to answer, because I have to say that I don't have, one scientist that was my biggest inspiration. And actually, what I've learned during my career and when I was younger, I've learned with amazing people. For example, when I was a PhD student, I was in Paris, and the director of my institute was a man called Axel Kahn, who died last year. But this man was absolutely incredible, because we had these conferences with all of the PhD students and the postdocs, and we were, maybe 100. So it was a big conference, and we were working on many different topics. At this time, I was working actually on HIV, but other people were working on cancer, and Axel was a cancer specialist. But he was there, and for each speaker, each PhD student or postdoc, he had a very, talented question. That was the question. So he was able to really understand everything, and I was impressed by his kindness and his willingness to teach things to the other people. And so I've learned that, the kindness and willingness to teach. And I was also I have to say that I had at this time an incredible PhD mentor, so that he was my director, and he taught me everything about molecular biology and how to handle things. And when I was desperate, for example, when I was like, oh, my PCR is not working, and at the beginning, actually, I spent like 6 months to try to succeed with my PCR, and he was like, this is only a technical problem, and a technical problem is not a problem. We're going to solve it, and don't be desperate because of that. You will solve it, and you'll see. Then you'll succeed. And he was right, actually. So I've learned, that tenacity. I've learned this kind of thing, that you sometimes you don't have chance, but you have to still focus and to still try, and you'll succeed at some point.
Yeah, fingers crossed.
Yeah.
The FSHD field will get there in the end. That's brilliant. Thank you very much, Julie. Is there anywhere patients can go to learn more about the work you're doing?
Well, actually, yeah, of course. We have a website where we put things ongoing. We have also a Twitter account. We have, and I have an email. So if patients want to talk with me, want to share things, want to be more involved, they can contact me, and I will do my best to help them or to explain things. I used to do that when I was in France. I used to talk with patients, like twice a year, and I still do it, actually. Among, I go in different regions of France, and to talk with different patients and to explain what we do, what are the major advances we've made during the last year, etc. So yeah, I'm always open to this kind of discussion. It's always a pleasure.
Brilliant. Thank you very much for your time today, Julie.
Well, you're welcome. I was really happy, and thank you for the interview.